Okay, today we're going to be looking at some axolotl embryos. So these are salamanders that only live in one lake still in Mexico. 20, 30 years ago, there was a whole handful of lakes that they still lived in, but pollution and other things have made them more of an endangered species. We ordered these from the axolotl colony at the University of Kentucky, where they have been breeding axolotls in captivity for a very long time. And so these animals have been used in regeneration studies for well over 100 years, easily. Papers published in all the major scientific journals about axolotl research. So it came in this little box in the mail, okay, overnight shipping, and I'm doing the unboxing here. The embryos have been put in a padded bag. I am going to have to cut this so that they don't get jostled too bad during shipping. This is just the outer bag. On the inside, we have how the axolotls are normally shipped. So these are axolotl embryos. 50 wild-type embryos to Dr. Christensen. And all those little balls floating around at the bottom are fertilized eggs. These are at the earliest stages of cleavage. So there are not very many cells. They are single cells, or they've divided into 2 blastomeres, or 4 or 8, or a morula stage. Very few of them would be to the blastula stage yet. How do I know that development hasn't been happening yet? Because of this. In the bag, there's some ice. So pretend that you were a frog or a salamander and you were an egg that was laid into a pond. If it was, say, during the spring, when the ice was melting, that water would be so cold, still having ice chunks in it that are all melting, the embryo wouldn't develop at all. And so we've kept these cold so that we can watch them from the earliest stages. So the water that they've been kept in and shipped in, I'm going to mix with water that has been aerated. Aerated means the lid was never on tight here, and air has been able to bump into the water molecules. This was chlorinated water from the tap. Chlorine kills axolotls, kills any fish that you would put in there, or these are salamanders, but it kills any pet that you would try to grow. So we need to get rid of the chlorine. It turns out that simple exposure to the air molecules eventually leaches out the chlorine, which is why people have to constantly measure their pools and see how much chlorine is there and add more chlorine, because the chlorine actually sort of just binds to the air and leaches away. So this has been dechlorinated. So I'll just pour some of this in here. This large container is called a carboy, usually 5, 10 liters, so we have some really big ones of these. And I'm just going to go ahead and allow these to mix. Now if this had been first thing in the morning, these have actually been sitting, they arrived earlier today. If this had been first thing in the morning, I would have let the bag sit in the water for a long time to make sure the temperature was exactly the same of the water that's in the container and the water that came shipped on ice. But I believe that it is warmed up enough that this water is not a significant temperature difference from the water that is sitting here at room temperature. And so, on your marks, get set. When I say go, these guys are going to start developing. Okay? Go. Those tiny little black and white balls that are swirling around in this water have enough yolk to turn into something that looks like a tadpole. So these eggs have been fertilized. They are zygotes. They are blastulas. We are going to see what stage of development are they in by putting them under a microscope.